So research is now having big strides that we think will help in diagnosis. For example, we are developing assays in CSF, in blood, in nasal swabs that enable us to detect the ability of this protein called α-synuclein that we know plays a major role in disease, and we're looking at the ability of this protein to seed the aggregation of α-synuclein. And this is very important because this can distinguish, it seems to be able to distinguish very nicely someone with Parkinson's from someone that has another form of a synucleinopathy even. So there's big hope there. Then imaging biomarkers are also an area of tremendous research right now. If we can, instead of having to look at the brain only post-mortem, if we can now give a staining agent that will go into the brain, label the α-synuclein inclusions, and we can detect this in the patient, in the person while they are alive, like we can already do for Alzheimer's. This would be a tremendous advantage as well. Genetic tools have evolved tremendously also in the past decades, so we now can sequence whole genomes very rapidly and get a lot of information. So the challenge is going to be then how we're going to combine all of this information. But there's hope because this type of approach is already being done in Alzheimer's disease, that we can combine different sources of information from different markers and then have a better measure for where we are in disease, in the disease and in the disease progression in particular.